Well, that's meiosis for you. Oh wait, was I too fast? Okay, let's take a step back. Well, first off, meiosis is a cellular process that is used to turn diploids into haploids. Meiosis has many stages and is also similar to mitosis. So before we get started, let's go over what the cell has to do in preparation. Before a cell undergoes meiosis, it has to go through interphase. Interphase has 3 stages: G1, synthesis, and G2. In G1, the cell grows more organelles and gets bigger. During synthesis, DNA replicates. In G2, the cell continues to get bigger in preparation for meiosis. The first stage of meiosis is prophase 1. In prophase 1, the nuclear membrane begins to disappear. Spindle fibers begin to grow in between the centrosomes. Chromatids begin to turn into visibly seen chromosomes. In one chromosome, there are 2 sister chromatids. Once the chromosomes are visibly seen, a small process called synapsis occurs. In synapsis, tetrads form. Homologous chromosomes in a tetrad cross over each other to form a chiasma. Once that happens, pieces of the chromosomes are exchanged. That's the end of prophase 1. Now we begin metaphase 1. In metaphase 1, the tetrads move to the middle to align themselves at the equator. They align an independent assortment. This is the key to genetic diversity. Now begins anaphase 1. This is where homologs move to opposite poles of the cell. However, the sister chromatids remain attached at the centromeres. Once they reach each pole, telophase 1 begins. The nuclear membrane begins to reassemble, the spindle fibers disappear, and cytokinesis divides the cell into 2. The result is 2 haploid cells, and now begins the second part of meiosis. Here's prophase 2. Similar to before, the nuclear membrane disappears, and the spindle fibers form in between the centrioles. However, synapsis does not occur again. Once interphase 2 is finished, metaphase 2 begins. Once again, the chromosomes align themselves at the equator of the cell. Now it's anaphase 2, where the sister chromatids separate and move to opposite poles. At the equator, the sister chromatids separate. And lastly, telophase 2. Just like before, the nuclear membrane reassembles, spindle fibers disappear, and cytokinesis divides the cell into 2. This time, however, the chromosomes decondense back into chromatin. Now we're left with a total of 4 haploid cells. And that's meiosis.